Hello and welcome to this video on Peptidoglycan, an important bacterial cell wall component and a target for antibiotics. If you're being treated for a bacterial infection, you're likely to think of something like penicillin. That is because penicillin is one of the oldest known antibiotics. It is a product of mold and it works for some bacteria, but not others. What makes penicillin effective, or perhaps it's better to say what selects for penicillin and similar classes of antibiotics, is the presence of a thick peptidoglycan wall. These bacteria are known as gram-positive bacteria. By contrast, gram-negative bacteria have a thin to almost non-existent peptidoglycan wall. And this is primarily what things like penicillin affect. So based on that, it's fairly clear it's kind of important, and not just as an antibiotic target, but purely for a bacteria's functionality. Anything structural to this extent is important, unless it is an extra part from the flat pack, which is either critically important or goes to some mysterious other dimension part of the thing you are building, and you will never find where it's meant to be. Let's start with the basic bacterial structure. Okay, maybe that's too basic. Let's start with something a little more detailed. Like all living organisms, bacteria have a nucleus full of DNA. Now, unlike the more familiar cell, it's not an actual nucleus that you would be more familiar with. It's really just a giant loop of DNA. Occasionally you'll also find plasmids, which are smaller circular bits of DNA that are separate from the main DNA. This can be acquired DNA or genes for antibiotics as an example. This can be acquired in a rather violent and aggressive manner. It's kind of amazing how much of microbiology or biology as a whole involves phallic objects, piercing into things, injecting, and then depositing a surprising amount of genetic material or similar markers. And next we have the limited organelles, such as ribosomes. These are needed to make proteins. The bacteria don't really have a lot of internal organelles other than these, and it's generally described as a membrane-bound organelle, if it is something more extensive. We now have the cell periphery. It is made up of an inner plasma membrane. This is a place where portals of entry, receptors, transporters, membrane-bound proteins, and so on are found. This membrane is functionally similar to that of a human cell, but structurally somewhat distinct and different, but for the most part you can think of it as a bacterial equivalent. The next outermost layer is the peptidoglycan cell wall. This is a major structural element and provides both strength and protection to the bacterium. While not a perfect analogy, imagine the way a crab has an exoskeleton. It is kind of similar in the structural support it provides, and to a certain extent the protection. Beyond this, you may also find a capsule or slime layer. Of interest in this video is the peptidoglycan cell wall. As mentioned earlier, in gram-positive bacteria it is thick. The gram-positive name references a gram stain, which shows up thick and bright when staining for these bacteria. Gram-negative bacteria are negative because, well, unsurprisingly, when you stain, the bacteria peptidoglycan wall is thinly stained or not at all, which is why they are called gram-negative. These features are important as it is part of the identification of bacteria. You can get an idea based on just the shape, size, division, peptidoglycan presence, media it grows on, and colonies it forms, and you can make a fairly accurate guess as to what the bacteria is, without having to go through the extra testing or even as far as genetic testing. If there's no stain at all showing, either it's not a bacterium, or you screwed up and forgot to add the crystal violet part of the gram stain. Alternatively, you were just too keen with your alcohol wash, but most likely, if no stain is showing up at all, you made a mistake. It's just the way life is. Other than a identification tool for different bacteria, we have to ask, why is peptidoglycan so damn important? To understand that, we need to look at what it does in the bacterial cell wall. We did say it's an important thing for structural strength, but there's more to it. Peptidoglycan is a polysaccharide. It's a fancy way of saying long chain of individual. In this case, we're talking about a combination of sugars and proteins. Each of the individual units gets put together via those ribosomes, and essentially it's like a giant chain, except in this case it's more like a chain link fence, where you have both horizontal and vertical linkages. Now, the horizontal, the polysaccharide part of this, is a sugar, whereas the vertical, that is the up-down connections, are amino acids. This is what allows it to form a mesh-like structure. The difference between this and say something like gelatin is that gelatin is just protein, but peptidoglycan is a combination of protein and sugar. In terms of the sugar component, we have two alternating residues. One of these is the beta-1,4 linked N-acetylglucosamine, or NAG, and the N-acetylmuramic acid, NAM. The NAM is itself linked to the oligopeptide chain, that is the protein part of this, and that protein chain is only made up of 3 to 5 amino acids. This means that the peptides that link the different parts of the sugar chain are actually quite small and keep it tightly bound together. They are, in a manner of speaking, very similar to the mortar in a brick wall. They keep the courses or layers of bricks connected together, and seated at least mostly flat. Because of that though, that does allow peptidoglycan to serve a very significant structural role, and it gives it that strength. Part of that strength is also in counteracting the osmotic pressure within both the cytoplasm and the extracellular fluid's osmotic pressure. The peptide chain in more detail is going to link itself from the top to the bottom peptidoglycan, or at least more accurately, the polysaccharide chain that makes up the peptidoglycan. Now, other than structural strength, you need this because, well, biology has this weird thing of either trying to stop or amplify osmosis, and for good reason. If osmosis was left uncontrolled, you would constantly have cells just lysing in your body from water rushing in, or dehydrating and collapsing from water rushing out. You want to have it happen very quickly, but you also don't want it to happen all the time. So now we know why it's strong and to a certain extent what it's made up of, we now need to look at peptidoglycan and see why it's important, or arguably more important than cats and wine to middle-aged and lonely people when we talk about bacteria. Its importance is not to be underestimated because it is an exceptional target for antibiotics. However, using an antibiotic that targets peptidoglycan against bacteria without that kind of cell wall, that is without a thick peptidoglycan layer, will be almost useless. To be clear about this, it will have an effect. It'll kill any bacteria in your GI tract that have that thick peptidoglycan wall, that gram-positive bacteria, but it won't help with the actual infection causing problems. Wiping out some bacteria in your GI tract will lead to a bad case of diarrhea. It could be worse, but no matter what happens, it isn't fun. The mechanism behind most of the antibiotics, I'm talking here mostly about penicillins that target peptidoglycan, are largely similar, although newer medications may change this with time, and there is some peculiarities to different drugs, antibiotics that are out there. First, we have its effect on the actual production of peptidoglycan. You need certain enzymes, specifically the DD transpeptidases, DD, in order to have peptidoglycan be made. Without this, you just don't get the production of what you need. Mostly we're here talking about the short linkage proteins. Penicillin in itself will block these proteins from being formed. If the penicillin doesn't outright block the formation of the proteins, it will instead prevent the proteins from forming bonds between the layers of the oligopeptides or the polysaccharides that make up the peptidoglycan. This would be like, for example, taking away the mortar in a brick wall. You can stack bricks up only so high before they lose their structural integrity. And then of course we have the fact that, well, bacteria like to split and form new bacteria. And so if you can't reliably create a solid cell membrane, and a cell tries to divide, well, you're going to have problems with that cell basically lysing itself. It's also only a matter of time before bacterial germ theory deniers make claims about antibiotics and gram-negative bacteria on par with conspiracy theorists, and claims about steel beams and jet fuel. Of course, it's not the only thing that penicillin and similar drugs can do for gram-positive bacteria. You can affect other steps in the peptidoglycan synthesis process, and some, for example, the topical antibiotic bactracin, targets a particular pyrophosphate, and this in turn is then intended to prevent the production of a large part of the peptidoglycan synthesis process, or at least the production stage. We also have lysosomes. Lysosomes are, well, essentially enzymes that will break down things. Now, the most potent form, or most common form might be better to say, is actually found within your tears, and it's part of your innate immune system. That is the part of your immune system that's able to respond to everything, just it's not great at every job, it's a jack of all trades. What it does is break down those beta-1,4-glycosidic bonds that make up the peptidoglycan on a horizontal way, that is between the two kinds of sugars or polysaccharides that make up peptidoglycan. This is one reason why lysosomes are more effective and more important in gram-positive bacteria, and significantly less so in gram-negative bacteria. Also, the fact that gram-negative bacteria tend to have a lipid polysaccharide layer that's a combination of sugars and fats rather than a peptidoglycan layer. This might be why unicorns' tears are meant to be the cure for this, that, or the other. Your desperation and despair failing to find them leads to you crying and thereby producing your own cure. There's also the susceptibility of certain bacteria to degradation due to exposure to antibiotics. As a general rule, if a bacteria is exposed to an antibiotic, you don't necessarily need to give the full dose, so to say. Theoretically, the inhibition of certain production pathways within the bacteria could, and understand this is a very hesitant statement, inhibit synthesis of peptidoglycan enough that the bacteria itself is weakened and then more easily destroyed or degraded by lysosomes. So you could theoretically use a combination of things rather than just antibiotics alone. We mentioned earlier two important things. One is gene exchange, that's the phallic reference and the whole idea of depositing genetic material. But the other is bacteria surviving exposure to antibiotics. That is, if they acquire resistance to antibiotics to gram-positive bacteria through exposure and then they transfer it, you can find yourself fighting a bacterium that is resistant. This also leads to the problem with antibiotic resistance. There are a surprising number of mutations that can occur, but a lot of them are for transpeptidases, and this will reduce the ability of most penicillin-type antibiotics to actually interfere with the production of those cross-linking proteins in the peptidoglycan wall. There's also another concern, particularly in the mycoplasmas, where because they essentially don't use one particular kind of, let's say, enzyme, peptidoglycan isn't as vulnerable, and because of that, penicillin doesn't really work very well against them. There's also several bacterial species that have been able to mutate or develop modifications in the way they essentially structure the polysaccharides, and this has led to them being even less vulnerable again to lysosomes. This is why you should not cry, it breeds antibiotic resistance. But also, it's one reason why giving antibiotics to somebody who isn't infected, or at least the pathogen isn't a gram-positive bacteria, can be a problem. The gram-negative bacteria, that is the bacteria that shouldn't be affected by antibiotics in this form, can then pick up and acquire these traits. They can then pass them on to gram-positive bacteria. It's rarer, but it can be done. This is why peptidoglycan is important and how it is used by the bacterial cell. It's defeated with antibiotics, but it's also how the arms race between pharmacology and pathogens is being pushed back with antibiotic resistance particularly, and how even some of our own innate immune system is kind of several generations old now when it comes to defending the human body from certain bacterial pathogens that are gram-positive. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing, and please do post any comments, questions, or suggestions you have below.